Today, we're diving into a kind of microscopic treasure hunt. Our mission, should we choose to accept it, is to pull the literal blueprint of life, yep, DNA, out of a regular sample of human blood. We're going to do it with some clever chemistry and a bit of spinning. So let's get into it.
So how on earth do you go from this, familiar red liquid to the incredibly complex blueprint that makes you, you? It honestly seems like it should be magic, right? But it's not. It's just pure, really elegant science. Let's break down how this whole puzzle gets solved. Okay, first things first, we've got to understand what we're working with. Blood isn't just red fluid. It's this super complex, kind of messy soup. It's packed with all sorts of cells, proteins, sugars, the whole shebang. And our target, the DNA, is locked away deep inside the white blood cells, hiding amongst all this other stuff. So our very first job is to clean house.
And that brings us right to stage one of our quest. We need to find the nucleus. Think of it like a heist. We have to break through the cell's outer defenses to get to the tiny little command centers where our genetic treasure is stashed.
So the process kicks off by adding a buffer, which just keeps everything stable. But then comes the really clever part. We add a specific detergent that's basically a key for only the red blood cells. It pops them right open. And since they don't have a nucleus, they're just cellular noise. We need them gone. Once they're all broken open, it is time to spin. And we do that separation using a machine called a centrifuge. The easiest way to picture what it does is to think of a super high-speed salad spinner. Seriously, it spins the tube so incredibly fast that it forces all the heavy stuff down to the bottom, while the lighter liquid stays right on top. It's a brilliant and honestly pretty simple way to separate things out. And after that first spin, what do we get? We get this, a teeny tiny white dot at the bottom of the tube. That is called the pellet. And I know it doesn't look like much, but that little dot is our scientific gold. It's a concentrated collection of all the nuclei from the white blood cells. We found it. So what about all that red liquid left on top? Well, that's called the supernatant. And it's basically cellular trash at this point, all the guts from the broken red blood cells. We just carefully pour it off and get rid of it. And just like that, stage one is done. All right, on to stage two. Purifying the DNA. We've got our nuclei, which is great, but the DNA inside is still all tangled up with a bunch of proteins. This next part is all about getting it clean.
So to crack open these nuclei, first we mix that little pellet into a high salt buffer. Then we hit it with a second, much stronger detergent. This one dissolves the membrane around each nucleus, releasing everything inside. And finally, we add a dose of really concentrated salt, which acts like a magnet for proteins, forcing them all to clump together and get away from our precious DNA strands.
Now here's a really, really important note. You'll see this in any real lab protocol. Do not vortex. A vortexer is a machine that just shakes tubes like crazy to mix them, and most of the time that's what you want, but not here. Here it would be an absolute disaster. And here's why. You have to understand just how delicate this whole process is. DNA molecules are these incredibly long, fragile strands. Shaking them violently on a vortexer is literally like putting a priceless ancient scroll through a paper shredder. You destroy it. So all the mixing has to be done by just gently turning the tube over and over by hand. It's all about precision. So we do another spin in the centrifuge, but this time way faster. All those proteins we clumped together, they're heavy, so they get slammed down into a new pellet at the bottom. But here's the crucial twist. Our prize, the pure DNA, is now floating in that clear liquid, the supernatant, on top. We carefully suck that liquid out with a pipette, move it to a clean tube, and toss the protein pellet. We have made it to the final stage. This is the moment we've been waiting for, the grand reveal. We've isolated the DNA, we've purified it, but there's just one tiny problem. We still can't see it. This last step is all about making our invisible treasure finally, magically, appear. So this is the final hurdle, right? The DNA is in there. It's clean, it's pure, and it's floating around in a perfectly clear liquid. It's kind of like trying to see sugar after you've stirred it into water. It's just gone. So how in the world do you make it solid again?
Well, the secret is actually surprisingly simple. It's alcohol. By adding some cold isopropanol, we totally change the chemistry inside that tube. See, DNA loves dissolving in our salty water solution, but it absolutely cannot stand being an alcohol. It's completely insoluble.
And now we get to the most satisfying moment in this entire process. You don't shake it, you don't stir it, you just take the tube and gently turn it upside down and back again, a few times.
That's it.
And then it happens. Just like that, right before your eyes, the invisible becomes visible. The DNA literally crashes out of the solution, forming these beautiful, stringy, cloudy white threads. That is it, the code of life right there for you to see. It is such a cool moment. But why do it? Why go through all this careful, delicate work? Because that single visible thread of DNA holds a literal universe of information.
You know, having a pure sample of DNA like this, it's the starting line for so much of modern science. It's what lets forensic scientists identify a suspect from a single hair. It lets doctors diagnose genetic diseases, sometimes before a person even has symptoms. It lets us understand the ancient history of how humans moved across the globe. Pretty much every major breakthrough in genetics starts with a process just like the one we just walked through.
So think about it. We've gone from a messy soup of blood to a tangible, visible thread of pure information. This process literally transforms the hidden into the known. And it really makes you wonder, with the blueprint of life itself right there in their hands, what secrets are scientists going to unlock next?